Welcome, listeners, to this podcast episode where we will dive deep into the innovative role of Dynabeads magnetic beads. But before we dive in, I just want to note that the products and the techniques in this podcast are for research use only and not for use in diagnostic procedures. Okay, now that's done, let's learn some more about our topic for today. So I'm your host, Claire Lund, and today we have a fascinating topic on the agenda: magnetic beads for personalized mRNA therapeutics. Now, this is a big and exciting topic, so I am thrilled to be joined by an expert from Thermo Fisher Scientific, a world leader in serving science. So please, could you introduce yourself and tell us a little bit about what it is that you do at Thermo Fisher?
Hello, thank you. It's a pleasure to be here. I'm Iwona Grad, an R&D scientist at Thermo Fisher Scientific. My current research focuses on simplifying the manufacturing of mRNA by solid phase in vitro transcription and purification using magnetic bead technology.
What a perfect background for helping us navigate this exciting mRNA topic. So, Dr. Grad, or is it okay if I call you Iwona?
Yes, please go ahead.
Oh, you're so kind. Thank you. Okay, Iwona, let's start with the basics. Can you explain to our listeners what is personalized medicine?
Absolutely. So personalized medicine, also sometimes called precision medicine, is an innovative approach to medical treatment that tailors healthcare to the individual characteristic of each patient. And those characteristics can be personal, genetic makeup, environment, and lifestyle. And taking them into consideration help to develop more effective and because they are more targeted, so they are more effective treatments. Standard medicine that we have today relies on the treatments that were designed with an average patient in mind. So they don't always capture those unique differences between individuals. While personalized medicines, on the other hand, is using advanced diagnostic tools and techniques such as genetic testing and molecular analysis to identify specific biomarkers and genetic variations that are unique to each of us.
Wow. So we're really tailoring our medicine now. Now I am even more intrigued. Okay, so Iwona, can you give us maybe a few examples or one example just to help us kind of contextualize this topic a little bit more?
Sure, there are many, but the most exciting are probably targeted therapies in oncology or genetic diseases. In oncology, we actually have had targeted therapies for a while now, such as HER2 inhibitors. However, with the progress of genomics, we can see that we are catching just the tip of the iceberg, meaning cancers harbor many more mutations than the actionable few that we are currently addressing. And those could be attacked with personalized cancer vaccines that are enabled by new tools such as mRNA Therapeutics. The mRNA Therapeutics also reveal new possibilities for genetic disorders, enabling protein replacement therapies and also gene editing.
Okay. Now, that is indeed exciting. Thanks for sharing the example. So, okay, now we have understood personalized medicine. Can you also then explain for us what are mRNA therapeutics and how do they work?
Sure. mRNA or messenger RNA is a type of genetic material that exists in every cell and instructs the cell to produce a specific protein. In the context of therapeutics, mRNA, which is produced through in vitro synthesis, can be engineered to produce proteins that can either treat or prevent disease when delivered to the target cell. Essentially, it works by giving genetic instructions directly to the cell, and the cell then produces the therapeutic protein that is needed.
Incredibly interesting. But can you illuminate a little bit why we need this advancement? Like, what advantages do we have by using mRNA therapeutics?
Yes, mRNA therapeutics actually are so exciting because they offer those numerous advantages over other types of therapeutics. The main one is that they can be developed more quickly than traditional therapies, and this is due to the rapid design and synthesis of mRNA once the genetic sequence of the target protein is known. A prime example is COVID-19 mRNA vaccine that was deployed in a record time. Then we have the mRNA's versatility. which means that it allows to encode for a full range of proteins, including those that are usually difficult or even impossible to produce by existing recombinant technologies. And then, of course, this makes it a much broader to treat different range of disease, from cancer to infectious disease and genetic disorders. mRNA therapeutics provide very precise instruction to the cell what type of protein to produce, and that leads to the highly targeted treatment with potentially fewer off-target effects. And also, unlike DNA-based therapies, mRNA does not integrate into the genome, which reduces the risk of long-term genetic changes and offers temporary expression, which sometimes is advantageous for certain applications and can enhance patient safety. Yes, additionally, the production of mRNA therapeutics can be theoretically scaled up relatively easily, which is important in times of pandemics or for widespread vaccination programs. So overall, when you look at mRNA therapeutics, their flexibility, precision, and rapid development capabilities make them such an interesting new tool in the box of the modern medicine.
For sure, definitely, clearly a lot of striking advantages there. And this all seems really positive, but are there any challenges that we see for mRNA therapeutics that we should be aware of?
Yes, of course, there are always some challenges, and the first one is most probably connected with inherent instability of the mRNA molecule itself, which can degrade quickly and requires advanced super clean manufacturing conditions to maintain this integrity. Then we have targeted delivery, which is yet another challenge. If we want the mRNA to be transported into specific cell type, this needs development of sophisticated delivery systems such as nanoparticles combined with molecules that are recognized by specific cell surface receptors. And still, manufacturing and flexible upscaling or downscaling of production to meet the clinical and commercial demands are still a challenge. And last but not least, the cost of production and ensuring consistent quality across batches can still be substantial. So we still have a few challenges to address before we take full advantage of those type of therapeutics in clinical practice.
Clearly, yeah. Well, let's hope we can overcome some of these challenges in the years to come. Okay. Can you also, you've already really helped us understand the personalized medicine aspect and the mRNA therapeutics. Can you now bring those two pieces together for us a little bit and tell us about personalized mRNA therapeutics and some of their challenges as well?
Yeah, sure. That's pretty simple. Personalized mRNA therapeutics simply means mRNA therapeutics used in the field of personalized medicine, means tailored to individual patient. And mRNA is perfectly suited for this since it's very easy to modify mRNA sequence, allowing for such customization to better meet the needs of individual patients. For instance, sequencing a patient's tumor might allow mRNA-based cancer immunotherapy for that particular patient. Then, to benefit the maximum number of patients, it is essential to produce hundreds or even thousands of different cancer vaccines or treatments in parallel. And this manufacturing process is different from the current vaccine-based approach, which relies on large bioreactors and downstream processing methods that are used for mass production. And this is a bit of a challenge because currently commercially available equipment and consumables for small batch processes are typically designed for research and development purposes. And those are not suitable for pharmaceutical manufacturing due to the lack of scalability, consistency, and compliance with good manufacturing practice, GMP. So, there are still some challenges. And if I were to summarize them, I would say cost is a big challenge. As always, the production of personalized therapies is often more expensive due to the labor-intensive processes and the lack of scale. So reducing costs while maintaining quality and efficacy is crucial, and this could involve innovation in automation, more efficient use of materials, and improved production techniques. Then we have turnaround time. To be effective, personalized therapies need to be produced and delivered quickly. That is especially crucial for oncology, as you can imagine. Here, innovation of the whole production process, but also streamlined supply chains are essential to meet those requirements. And then we have consistency and quality control. Assuring that each personalized therapy meets stringent quality standards is more challenging due to the variability of the starting material, for instance, a patient's cells. And therefore, using high quality raw materials is even more essential in this context. And of course, development of advanced and robust quality control. Yes, I would say this would be the main challenges that are still there, and overall they will require significant advancement in biomanufacturing technologies, like, for instance, modular and flexible manufacturing system. As I mentioned, automation. Also, it would be good to have integrated data management system that would allow to monitor and control the whole process in the real time.
Right. Well, that is quite a list of challenges. But Iwona, I heard that Dynabeads magnetic beads from Thermo Fisher Scientific can actually play a role in the production of mRNA therapeutics. Can you tell us a little bit more about that?
Of course. Dynabeads magnetic beads were developed over 35 years ago and remain unmatched in their monodispersity and high manufacturing quality. The beads have various applications in academic, industrial, and clinical settings. But the Dynabeads products that I want to tell you about today were specifically developed by Thermo Fisher Scientific for mRNA synthesis and purification. These are a new type of magnetic beads that are used in the production and purification of mRNA. And simply speaking, we have this unique approach to IVT by attaching the DNA template to magnetic bead surface that is followed by mRNA synthesis. And then this full-length template DNA is separated from the synthesized mRNA and can be further reused, while mRNA is purified in a very simple one-step process using another magnetic bead product.
Right. This sounds really intriguing. Okay, I want to learn a bit more. Can you tell us about why you would use Dynabeads magnetic beads to synthesize and purify mRNA, and also how they can address some of these manufacturing challenges for mRNA therapeutics that you spoke about earlier?
Yes. Our proprietary magnetic bead technology was designed to help to accelerate the commercialization of mRNA vaccines and therapeutics by streamlining the development and manufacturing process from benchtop to clinical bulk production. So using the same type of process on the small scale and the large scale. And this is enabled by high bead performance, scalability of the bead-based process, and security of the bead supply. If I went into the details, our unique approach to the IVT, which we call solid phase IVT, starts with increasing the purity of the synthesized mRNA from the beginning. And this is reducing DNA carryover to the further steps and decreases the complexity of the purification that is needed downstream. And this, combined with a simplified one-step magnetic bead-based purification, significantly reduces the manufacturing steps and decreases, therefore, the time and cost of the whole process. And these are some of the most important challenges in the field of personalized mRNA therapeutics.
Right.
Also, with our technology, the DNA template can be reused multiple times. And this minimizes plasmid preparation and reduces hands-on time. The bead-based process enables also very efficient purification of mRNA that results in a high yield. And the purification beads are reusable as well, which additionally reduces the production costs. So again, improvement in time and cost of manufacturing, which are those challenges that we discussed previously. Importantly, magnetic beads are inherently automatable. enabling this parallel processing of multiple reactions at the same time. And that is also crucial for the future of personalized therapies. Bead-based process is scalable, and that, as I mentioned earlier, allows the seamless transition from research scale to clinical and commercial production, ensuring consistent quality across different production levels. Additionally, the beads minimize footprint because they support modular manufacturing, and this can cost effectively match the required scale that we need at the moment. And lastly, Dynabeads magnetic beads also come in a GMP-compatible grade, which together with secured commercial supply enables consistent quality of the manufacturing process. So overall, Dynabeads magnetic beads are a unique tool for the development of fast, cost-efficient, consistent, and scalable manufacturing process. And this is due to their quality, high production yield, the fact that they can be reused, they give minimized footprint, and automation capabilities.
Wonderful. Wow. Okay, yes, as you said, pretty unique technology and so many advantages there in facing the challenges in this field. Great. So one last question for you, Iwona, and it's a big one, of course. Might require you to have a crystal ball. But okay, tell us what you think the future looks like for personalized mRNA therapeutics.
Yes, as you can imagine, I'm a big fan. Yeah. I think the future of personalized mRNA therapeutics is incredibly promising. And I think that those therapeutics will revolutionize the landscape of medical treatment. And this is, I know that we talked a lot about the challenges, but there is a continuous advancement in manufacturing and delivery technologies. Also, there are discoveries in genetic research, in bioinformatics, and with all this progress, I believe that mRNA therapeutics will likely become a cornerstone of personalized medicine. Moreover, nowadays we also have the integration of artificial intelligence and machine learning that will help in the development of delivery methods and optimization of the design of mRNA sequences, and that will also enhance this development of mRNA therapeutics and bringing it to the market. When I think about the promise of this mRNA personalized therapeutics, I think one of the most exciting prospects is the development of personalized cancer vaccines, where mRNA can be used to instruct the immune system to specifically target and destroy cancer cells based on the patient's tumor-specific antigens. So this is, it's really big. But of course, additionally, mRNA therapeutics hold promise for treating a wide range of genetic disorders by enabling the production of functional proteins that are missing or defective in patients, or of course, editing, gene editing by themselves. Another example is cell therapy. Cell therapy, which involves the use of living cells to treat or prevent diseases, which can be combined with mRNA technology to offer synergistic benefits. For example, mRNA can be used to program cells ex vivo before they are reintroduced into the patients, enhancing their therapeutic potential. And this approach is particularly promising for regenerative medicine, where modified cells can then repair damaged tissue, and also for immunotherapies, where engineered immune cells can be more effectively targeting and eliminating cancer cells. So, overall, the future of personalized mRNA therapeutics is very bright. And they have the potential to transform the way we approach the treatment of a wide variety of diseases. And they offer hope for more precise, effective, and individualized healthcare solutions.
Wow. Iwona, you really did leave us on a high note there. Plenty to be excited about for the future, it seems. And it really, truly is an exciting time for this field, it seems. All the rapid innovation of tools and technologies really looks set to significantly accelerate the development of mRNA therapeutics. Also, as you have eloquently explained for us today, just now, also that innovative technology, the Dynabeads magnetic beads, really seem to offer a unique and powerful solution for mRNA synthesis and purification. So with that, thank you so much, Dr. Grad, Iwona, for sharing your insights today. This was truly a fascinating discussion.
Thank you for having me. It's been a pleasure.
And to you, our dear listeners, I hope you found this chat as informative and exciting as me. And I thank you for tuning in today. Goodbye.